This is a video for the IB Biology course, topic 2.6. Here we can see all the understandings, applications and skills for this topic. It's quite a short one. It's all about DNA, RNA, and the structure of those molecules. So nucleic acids are the genetic material of the cell, and they're composed of recurring monomeric units called nucleotides. So here you can see a nucleotide composed of a nitrogenous base, a pentose sugar, and a phosphate group. So two different types of nucleotides that humans have, so RNA and DNA nucleotides. RNA, the difference is in the sugar with the ribose and the deoxyribose in DNA. The other differences are a different type of nitrogenous base is used for RNA, so thymine is used in DNA, but uracil is used in RNA, and the number of strands. So DNA forms a double-stranded helix, whereas RNA is single-stranded only. And this clearly shows the difference in structure. DNA, obviously 2 strands, double helix, RNA 1 strand, and then the difference in base. It's important to remember how to draw nucleotides and DNA. So this is how you should draw it if asked in a test, with a pentagon representing the sugar, a rectangle representing the base, and a circle representing the phosphate. And that's quite a universally known way of representing DNA. So here you can see that DNA forms into anti-parallel strands, which means that they are actually upside down, these molecules, although it might not look like it. So you can see here this strand on the left is the nucleotides are arranged in this group, with the 4 carbon pointing up over to the 5 carbon and phosphate, whereas over here the 4 and 5 carbon with the phosphate group is pointing down, and that's how they're arranged. The other thing that determines how DNA is arranged is that C and G bases, so your cytosine and your guanine, only bond with each other, and A and T only bond with each other. You cannot have an A and a G bonded together in a DNA molecule. So the structure of DNA was correctly proposed by Watson and Crick. So they constructed many models to quickly visualize, assess the viability of potential structures, so yeah, their efforts were guided by understanding of molecular distances and bond angles. On top of that, so here's just the evidence that led to the discovery of the structure. DNA was composed of nucleotides made up of sugar, phosphate, and base. DNA is composed of an equal number of purines and pyrimidines, and DNA was organized into a helical structure. So Rosalind Franklin discovered this with X-ray crystallography images, which directly led to the discovery by Crick and Watson of the structure. However, that data was not shared with her permission, so that caused some controversy. But that is everything for topic 2.6.